Hello everyone. Let's proceed with a continuation or let's continue our discussion on proteins. Let me just go over the previous presentation of slides during our initial meeting on proteins. So to recall, proteins come from the word proteus, meaning of first importance, and they're considered the most important of all biological compounds. So we already had an encounter with physiological functions, and we had started with structure, particularly your amino acids and the different classification of amino acids according to structure. We were basing on structure as to how amino acids were classified and how these structures relate to the properties. For this discussion, we'll proceed with how peptides are formed. So, I recall we already started and I left you with an activity. So, with this peptide formation, we're able to build our proteins. That's why we have different levels of protein structure and how these structures and peptide formation correlate with the formation of conjugated proteins, as well as the structure is significant in a sense how the denaturation process affects the ability of proteins, and lastly would be importance. So to recall, proteins are macromolecules with amino acids as their building blocks, and they are synthesized in the cell ribosome. So to take note, common elements among the three macromolecules are carbon, hydrogen, and oxygen. But for proteins, so we already have nitrogen, then these can bond with phosphorus or sulfur as well, just like lipids. So these are the different classification by physiological functions. And if you realize to think of it, your whole body uses proteins in different uses. And proteins may be classified by more than one function. Okay? So these proteins are versatile in a sense that your transport proteins correlate or collaborate with lipids, so complex lipids. Okay, your structural protein collaborate with contractile, so how your body moves without having to fall apart. Okay, and storage protein is in charge how storage protein collaborate with defense protein, that in situations such as starvation or in terms of low immunity, so how your body responds to such situations, and then how enzymes are able to collaborate with regulatory proteins, that in certain scenarios when you have some problems, so minimal problems in metabolism, so how enzymes cope up, since enzymes tend to be really specific, so every enzyme has a specific function. So that's why classifying proteins is not a one-way thing. And a little recall, so in terms of structural differences among the different macromolecules, so they always have one unit, one basic unit, or your monomer. So for carbohydrates, so your monosaccharide serves as your building unit. So if you have multiple units of these, so you form a chain, and that's when you have your macromolecule. So for lipids, so each fatty acid can bond with glycerol or sphingosine, forming your triglycerides or your non-glycerides or your sphingolipids. And lastly, for proteins, so your building blocks are amino acids. So usually when these amino acids bond with one another, so they form your peptides. And if they exceed. So I have this reading. So there are references that tell you amino acids reaching 50 in a peptide. So 50 or more, or some reference will say 51 and more, is already protein. So more or less, so just like vital signs. So that range is just a rough estimate as to how many amino acids there should be in a protein for it to be considered protein. Otherwise, it's called your polypeptide. Okay, so peptides are fewer than 50. So to answer, 50 or more is the safe answer. So 50 or more is amino acid in a chain, so that's proteins already. Okay, so in terms of structure, so proteins may be fibrous or globular, but inside these fibers or globules, each goes back to your amino acid. So recalling, so in terms of structure, you have your alpha carbon attached with amine group and the carboxylic acid group, and each side chain can distinguish one amino acid from another. Okay, and remember, the carbon bearing all these substituents is called your alpha carbon. And because of the presence of amine and carboxylic acid group, so your amino acids are considered Zwitter ions because it bears both equal positive and negative charge, usually at neutral temperature, neutral pH. So the pH at which when your positive charge is equal to the number of negative charges is called isoelectric point. So here are your alpha amino acids, which are the common amino acids. So there are 20, and each have their specific 3-letter and 1-letter abbreviation. And each of these have different side chains distinguished from one another. So you have alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, valine. These are the 20 alpha amino acids. So last time we classified them according to structure. Properties based on structure, we divided them accordingly. So we have non-polar. So usually you have methyl groups, alkyl groups rather. So all carbon and hydrogen as the side chain. Then we have polar, so meaning they are uncharged. So usually they contain several. So there's no definite group of side chain. So you may have your carbonyl, that's C double bond O, attached to another amine, but still attached to alkyl groups. And then you can also have OH attached to an alkyl, and of course you might have simply H, or hydrogen attached to or serves as substituent for your amino acid. Then we have acidic. So apart from the alpha carbon bearing amine and carboxylic acid, so for example in aspartic acid, so this is your alpha carbon bearing the main acid group. This one is the main amino group. So these two, so your CH2 and this one, this is still considered C double bond OH. So this is already a substituent. So this is already an R group. So meaning this defines the acidity of your amino acid, particularly aspartic acid. That's why they are called aspartic acid. Okay. Apart from that, so sulfur-bearing substituents such as methionine usually yield acidic substituent, or it makes the amino acid acidic. Then for glutamic acid, the same. You have, so this alkyl is ethyl attached with C double bond OOH. So this is glutamic acid. And lastly, when you have a phenyl, so if you recall, if you have a phenyl ring and you have nitrogen outside, so it leans to be an acidic in terms of pH. Plus you have a double bond, so you have tryptophan. Okay? Next would be, so apart from those, so you see similarities. So we have sulfur, the phenyl ring, you have nitrogen enclosed in the ring, as well as alkyl groups attached to phenyl. So we have phenylalanine, isoleucine, threonine, cysteine, histidine, and tyrosine, meaning these are acidic. And lastly, we have amino acids with basic property. So usually when you have nitrogen attached to alkyl groups without the presence of ring, so they are basic. So if you recall, so testing for presence of or composition of organic compounds, so when you have ammonia or ammonia gas, nitrogen containing, so usually they go on leaning towards basic pH. On the other hand, we can also classify your amino acids based on source or how these are synthesized. So that's when we classify them as essential, conditionally essential, or semi-essential, and non-essential. But to remember easier which one is essential, so we simply use the mnemonic. So private, then Tim, Hall. So I reviewed. So there's no A. So since there are just nine essential amino acids, to which we have to intake food rich in these essential amino acids to supplement to our body. So that's the major difference between essential and non-essential. Semi-essential amino acids are usually categorized as non-essential because the only time we cannot get them is when we have inborn disorder in metabolism, or when we are under stress, one of which when we're starving. So that's why we cannot synthesize these amino acids. So recalling, so your essential amino acids are, so P is phenylalanine, V is valine, T is threonine, T is tryptophan, so I is isoleucine, M is methionine, so H is histidine, L, so if you have isoleucine, you also have leucine. Lastly would be lysine. So looking at structure between isoleucine and leucine, so the major difference is one substituent. So this is a methyl group here. In leucine, it is right across your amine group. But for isoleucine, so the substituent is here, so away from, or it is away from the amine group. So that's the difference between the two. Okay? So usually when we think of amino acids and protein, we link this diet with heavy meat, not really heavy, but majority of the food containing protein is said to be meat. But actually, meat is not necessarily the only source of proteins or amino acids. Usually, these are in balance lang, no? Nga plants can also contain proteins. But in terms of economic comparison, so you will require more plants containing protein to extract about the same amount of protein we can get from meat. That's why it is more economical to obtain protein supplements or proteins from meat or dairy or even fish, because also there are amino acids not found in plants which are found in meat, dairy, or fish. Okay, so that's the reason why when people say protein diet, it is usually associated with meat or fish or eggs. So in terms of peptide formation, so that occurs when one acid group in an amino acid joins with the amine group of another. And this reaction is called your condensation reaction. So peptide formation is condensation, wherein we have water as your byproduct. And you also have if you're trying to um, break this bond, so if you recall, this peptide bond here is the linkage between the two different amino acids. So that's for condensation. But if you're going to break this bond, so that would be hydrolysis. Meaning water will break into OH and H to bind. So carboxylic acid group will be attracted to your hydroxide, while your hydrogen will be attracted to or attracted by the amine group. And this leads us to the different levels of structure of proteins. So one would be your primary structure, second is secondary, and lastly would be tertiary, and for some proteins, so they can reach quaternary structure. So for primary, this is simply your amino acids linking together. So it's a linear sequence. So two ends of this linear sequence might bear an amino acid and having free carboxyl, meaning your carboxylic acid group, or the other end would be your amine group. So the terminal, the end having a free alpha carboxyl group, so meaning if this is your carboxyl group, this is attached to an alpha carbon, which is represented here in this bead. So your alpha carbon is found here. So this carboxyl group is called your C terminal. So your carboxylic group again attached to this amino acid. So this amino acid is called your C terminal or C terminus. On the other hand, if your amino acid has a free alpha amino group, this amino acid is your N terminus or N terminal. So that's for primary structure. This is 2D. If different linear sequences link together in certain bonds, which they form either sheets or helix, that's what we consider your secondary structure. And these tend to be repetitive. So this is usually repetitive but short range. So you can either have alpha helix or beta sheet protein structure. And these two can exist in one structure, which we then call your tertiary structure. Okay, so as much as proteins replicate and bind or fold themselves, so they can actually also help other proteins to fold into different conformation at their favorable sequence. So when you look at this structure, so you have a combination of alpha helix. So the blue one is alpha helix, and the blue one with the coil is alpha helix, And the one with a flat conformation is your beta sheet. So when we see this, so here, coil, this is alpha helix. So consistent nga pattern, that's alpha helix. So you might also have another helix here. This one is also a helix. Even this one. As well as here, and here, and here. So meaning, repetitive but sagol na siya. For this one, so this is considered a sheet. Even here, since there is no repetitive pattern. But when we reach here, so kani siya, kani, this is already alpha helix. And the others, so kaning straight lang, planar, that is already beta sheet. So tertiary structure is considered three-dimensional conformation already of your protein molecule. Okay? And lastly, for some proteins, they are able to form quaternary structure. So polypeptide units can clump together. So they form a lump or clamp or clump something. So basta globular, usually globular, then that's your quaternary structure. So if this clump mass, compact mass, is already aggregated with other, let's say, ions or other units, then you can already have your conjugated proteins. And one perfect example of proteins having quaternary structure would be, which is also a conjugated protein, would be hemoglobin. So if you notice the structure of hemoglobin, you already have the presence of heme bearing the iron ion. So iron is a non-amino acid, thus this is your prostatic group. Okay? So all proteins that contain non-amino acid portion is considered conjugated protein with a prostatic group. However, not all conjugated proteins have prostatic groups. And one example for that would be collagen. So the reason why it's considered conjugated is because if you look at the sequence, so you have glycine and proline, which are alpha amino acids. You also have hydroxyproline, which is another amino acid. But what makes this conjugated is that your glycine and proline are alpha amino, but hydroxyproline is possibly derived from proline. So meaning it is conjugated. Okay? And this is usually forming fibrous structure. So thus you have your collagen fibers. Okay? So this is your no prostatic but conjugated proteins, while hemoglobin has prostatic group. And apart from heme as an example of prostatic group, so you also have other classes of proteins containing different prostatic groups. So for glycoprotein, simply look for the presence of a saccharide. And then for hemoprotein, so you have hemoglobin or could be myoglobin. So hemoglobin can also be classified as metalloprotein because it contains metal, so in the form of iron. So apart from metalloprotein, we have lipoprotein, your complex lipid. So you have your good and bad cholesterol. So they are lipid carriers or transporters, and your VLDL. And then of course, you have nucleoproteins. So you look for the presence of nucleic acid. And lastly, for phosphoprotein, so you look for phosphate esters. One example would be so casein found in milk. So these are just some, or these are the major classifications of conjugated proteins as well as their prostatic groups. So it's really quite very obvious, no? So glyco containing sugar. Hemo contains heme. Lipo contains lipids. Metallo contains metals. Nucleo contains nucleic acids. And phospho contains phosphate esters. Now, since proteins are versatile and used in a lot of bodily functions, they are also prone to denaturation. So this process is simply the loss of functional shape of your proteins because of different agents. Usually they are called denaturing agent. So if we've always mentioned proteins are sensitive, so we can also tag them as OA at some extent. So proteins can alter or can lose its functional shape in the presence of heat. So it can also be in the alteration of pH, whether it is more acidic or more basic. In the presence of heavy metals, even alcohol, or even detergents, salts, reducing agents, and the like. So, apart from denaturation, proteins may also lose its functional shape due to errors, which we usually say protein misfolding. We refer as protein misfolding. So, protein misfolding can occur due to different factors such as one, genetic mutation. And this is usually not much of control since it happens within the DNA of the organism. Next would be environmental stressors. So this is related to denaturing agents such as temperature, heat, or so much coldness, then changes in water levels or salt levels in the body. And of course, protein misfolding can happen due to aging. So if we think about it, when people age, so their metabolism rate can either can either stay the same, or it usually declines. So the ability to regenerate new cells will decline. That's why when a person is ill, so age usually is a factor of how fast that person can recover. Or when the person has a disease or disorder, the chance of recovery might not be 100%, but it can also depend on the state of the how severe or how mild the person's disease or disorder is, or how fast can the body of the person cope up. Okay. So some examples of denaturation. So one example, so in terms of changing pH, would be curdling of milk. So if you have milk and you add acidic lemon juice, or in the lab when you have milk you add acetic acid, glacial acetic acid, you'll be able to separate two different phases. So you have the solid phase. So here in the picture, the one on the spoon is the curd, and the liquid phase will be the whey. So the one on the container. So the surrounding liquid is whey. So aside from that, so the effect of temperature that alters the protein structure or shape would be cooking eggs, for example. So if you hard boil the egg, so the moisture or the water inside of the egg is water. The moisture there is trapped, so masolidify mong egg. And then this will indicate the yolk will also harden soon enough due to the presence of heat. Okay, so when the water is trapped and then your proteins are denatured due to heat, so the yolk will also harden, so you have protein denaturation by cooking egg alone. Apart from that, another situation of protein denaturation would be when a person has a fever. So there's always a rationale behind why should a person put effort in decreasing the temperature So during fever, particularly in the forehead region. And that's because of the possibility of damaging heat-sensitive cells, particularly in the brain. So, di ba, if a person is sick and the temperature exceeds, say, 40 degrees Celsius, so that person is prone to convulsion, and your cells in the head region, the brain region is at risk. So, that's why when a person has a fever, papilit dayon, cool fever, or some will dip face towel or cloth in cold water and put it on the forehead. Okay? So, even though fevers are considered beneficial in terms of inhibiting infections or inhibiting reactions that could trigger infection, further infection, since ang fever ni occur kaya na may infection. So it can also possibly damage other cells which are sensitive to heat. Then apart from that, so amino acids are linked to production of other biomolecules usually. So we have enzymes or hormones, and one of which are aromatic amino acids. They are precursors of your neurotransmitters. So we have three particular amino acids which are aromatic. So we have phenylalanine, tyrosine, and tryptophan. So they are used to create or to produce neurotransmitters. So here is a diagram showing us the uses of proteins in the human body. So from head to toe, proteins are everywhere. So from the signaling, so in the brain to other cells and back, so proteins work as communicators. And the immune system, they fight infection and even give or activate so certain molecules to fight whatever foreign invaders to the body might possibly exist. Then, in terms of structural, so your muscles contain proteins, how your muscles move, and proteins in the blood, so your carriers of oxygen, and how these are regulating or how your proteins regulate the levels of liquid in blood. Then your structural protein, so together with the muscles, your cytoskeleton, and even your hair to your nails, why they are, how they are there and how they exist without having to be, even though they are sensitive, but how they cope up, no? So, di ba, hair are really quick to grow for most people. Although some, there are certain reasons why it takes time for their hair to grow. But usually, people have fast growing hair and nails. And of course, your cell membrane, so proteins are there to integrate different cells. And lastly, so if we recall, we have enzymes which are a form of globular proteins. Now, to appreciate more the presence of proteins in the body, so diseases are there to remind us the essence of correct protein regeneration or reproduction. And what are the possible diseases which could be resulting from protein defects? So first off, we have sickle cell anemia. So I know you're very familiar of anemia, and there are different types of anemia. So first would be iron deficiency, which is more common, and the other will be sickle cell. What happens here is looking at the sequence of amino acid in hemoglobin, we notice that the sixth amino acid should be glutamic acid. But if this amino acid is replaced by different, so say for example valine in place of glutamic acid, your blood cell cannot form a normal shape. It forms crescent shaped cell, and therefore it cannot accommodate enough amount of oxygen to carry throughout the bloodstream, and this sickle cell often hardens and get stuck in the passageway of blood, so meaning it can form blood clots. That's why it has to be treated. Another or other diseases caused by protein misfolding perhaps would be, so one is Alzheimer's disease. It is a form of dementia. So it is characterized by progressive memory loss and a decline in cognitive function. So people with Alzheimer's usually gradually begin to forget simple things, but it can also reach a point in which that person can no longer recognize even themselves, or that person can no longer determine it's time to eat, it's time to sleep. So wala. That's because the brain can no longer function well. So on the other hand, for Parkinson's disease, so still this is in relation to protein misfolding. So these usually affect people beyond 50. But if the person is affected, wa pa siya kaabot 50, so it is usually termed as early onset. Though possibly earlier or younger, that person already showed symptoms, it's difficult to determine because when we say cognitive function decline, so understood naman nga, it is normal to forget sometimes. But if it happens frequently, then there must be something wrong. Because our body is busy performing a lot of functions. So sometimes memory, the brain cannot cooperate well. But if your basic functions are already compromised, so there is something wrong. Even to the point of forgetting how to properly walk, how to properly tell the time, that's already something. Apart from these two, we have your mad cow disease and we have transthyretin disease, usually associated with your internal organs. So for mad cow disease, these are involving mad cows or cows, but it is a disorder. So disorder caused by the misfolding of disease or misfolded proteins which they get from contaminated food containing misfolded proteins. Then these cows lose the proper way of moving. So just like how Parkinson's affect or how Alzheimer's affect humans, so they can also affect the cows in a way that cows cannot think properly. Then, is mad cow disease transmissible to humans? Yes, they are. But is it preventable? Definitely. So that's why we need to screen the animals. So when we have animal-based food, there is actually a celebration of safe meat, no, safe meat month. I think I was able to come across a motorcade. So it is usually held by companies or factories producing food products involving meat. Because through assessment of safe food, then we can prevent diseases which are related to proteins. So just like this one. Then for Parkinson's disease, so these are just some of the symptoms which can tell whether a person has Parkinson's. So as my mother usually describes it, more siya pang elite class nga disease because this is linked to the lifestyle of a person. That's why it's important also to keep ourselves kanang awake, alive, alert, enthusiastic, something like that, so that our cognitive function can also be exercised. So as much as our body physically needs exercise, we also need to have mental exercise. Because as we grow old, so our body must not stop thinking or must not stop exercising how to decide on things nga para dili ta what we call matayan. Because sometimes these are some causes of protein degenerative disease. So when we say degenerative, it involves decline in the ability um, to perform. So going back to the structure, so we learned that we have fibrous and globular proteins. And inside these, we have the presence of long chains of amino acids. And that leads us to our next topic, which is enzymes. So enzymes are considered biological catalysts. So in other words, they speed up the reaction of those going on inside the body without themselves going and undergoing any change. So enzymes cannot be consumed in the reaction, but they help speed up the chemical reaction. And they are generally made of globular proteins. But there are enzymes made of RNA, which we call your ribozymes. And they are physiologically specific. In the simplest manner of describing enzymes, one enzyme has one function. An enzyme was made for one specific function wherever it is found. But they can be reused. They can be reused depending on the compatibility of their function towards the certain reaction. Okay? But for this lesson, so we're going to go over, we'll no longer proceed with kinetics since it is beyond our scope, but we will classify the different enzymes. Then we'll have an overview of the terminology, which will be used also later on when we discuss metabolism. Then we'll talk about the importance of enzymes, the specificity of enzymes, what are the some of the models. Since enzymes are specific, we cannot name each structure because there are millions if not billions of enzymes existing. So we'll just have some models to represent how enzymes work, and lastly, associated disease. So that will be for our next topic.